So the clinical trial in the newborn kids is a trial of 3 centers. I mentioned the centers already: Seattle Children's, St. Jude is the lead, and UCSF. The lead investigators presented 8 patients who had been accrued in a period of under 2 years from the first 2 institutions that enrolled, and the results were presented at the American Society of Gene and Cell Therapy in May of last year, and they were extremely compelling. All patients were either completely had reconstituted their immune system and were doing well and were home, or were on the road to that quality recovery in terms of getting over infections they already had, beginning to reconstitute the immune system, and heading towards the outcome of the patients who've been followed for a longer period of time. So those results will be updated in a major medical publication this year, which has already been accepted for publication. Extraordinarily compelling results with an excellent safety profile, including the safety associated with the conditioning regimen, which is a low-dose busulfan, so a non-myeloablative dose of busulfan, which is a chemotherapeutic agent and has not been associated with infections, has not been associated with the need for transfusions of platelets or red blood cells. So very well tolerated. And the second trial was published in Science Translational Medicine in 2016. Again, that's the trial at the NIH under Harry Malik. 5 patients were published at that time, and the results were comparable. What's interesting there is that 2 of the patients had been treated early on, and they had at that time anywhere from 2.5 to 5 years of follow-up. And so what's important there is that there's been no incidence of abnormal proliferation of cells or frank leukemogenesis in these patients, which is a problem that had plagued earlier generations of the viral vector used to transfect the patient's hematopoietic stem cells. Because we just licensed the product in August of last year, we haven't yet had the opportunity to get in front of the FDA. So obviously there have been many FDA interactions on behalf of the NIH as well as St. Jude, but these have been related specifically to the protocol and the conduct of the study. We hope to get in front of the FDA in the first half of this year with a PreID meeting to discuss critical issues in the submission, in the BLA submission, such as patient number and follow-up for each patient that the FDA will require. Based upon Strimvelis, which is an ADA SCID product that was approved, gene therapy product that was approved in Europe, we see numbers being as small as 12 that the FDA would require for approval. So right now, as I said, 8 patients have been published. We certainly believe that we're going to have to process some cells in our own cell center here in Worcester, but the number should be overall quite small to achieve at least conditional approval, with a follow-up that we hope will be as short as 2 years. So that would lead to BLA filing somewhere in the 2021 timeframe. So in terms of getting sufficient patients, so Seattle Children's just came on board actually in August, the same month that we licensed in the program. So we haven't yet seen accrual there, but we believe, based upon the reputation of the center, that that they will add incrementally to the accrual that we've seen so far. Certainly the great results that have been seen have helped with accrual since the publication or the presentation in May of last year, and we believe the publication this year will get the word out further. So we think that parents and patients, when they're independent, are certainly willing to travel to these centers for therapy. St. Jude, being a very important charity, is able to bring in patients at low or no cost from outside the U.S. as well. So they have accrued patients, ex-U.S. patients, into the trial. So we believe that accrual will accelerate, and that we should not have any problem completing that trial once the investigational new drug application is transferred to us and we start processing cells here. In terms of logistics, thanks to the CAR T world, the logistics for shipping frozen cells and getting them reliably, transducing them in a center and shipping them reliably back to that same patient have been worked out. There are a number of organizations that are able to take care of that. We contract with Traxcell, which is a very well-known vendor in the field, to ensure chain of custody of those cells, that every step along the way that the patient is identified and the cells do go back to the same patient. We deal with premier carriers in terms of cold chain, so that cells are shipped frozen, and that frozen state is maintained so that the viability of the cells is intact for the entire journey. In terms of commercial, we have certainly run the numbers, and we do believe that the combination of the newly diagnosed patients as well as tapping into this reservoir of patients is a very attractive commercial model in terms of revenues for a company of our size and even a company much larger. Furthermore, we are designing the center that we're in now is built out for U.S. cell processing. And fortunately, we have another half of this facility that's not built out, and we plan on building it out to European specifications so that we actually can process cells from around the world, which will obviously double or even triple the market size for Mustang.